The overall goal of this protocol is to quantify small non-coding RNAs on purified lipoproteins.
This protocol can help answer key questions in the lipoprotein field, such as defining lipoprotein small RNA signatures in health and disease, as well as advancing lipoprotein small RNAs as disease biomarkers or cell-to-cell messages. The main advantage of this technique is one can analyze highly pure lipoproteins for small RNA changes in disease or experimental conditions.
The implications of this technique extend toward the therapy of cardiometabolic diseases because extracellular RNAs are a new class of disease biomarkers, and lipoprotein small RNAs hold exciting potential as drug targets in future studies.
Though this method is designed for HDL small RNAs, it can also be adapted for very low density and low density lipoprotein small RNAs.
Generally, individuals new to this method may struggle because of lack of proper equipment and expertise in preparing samples for density gradient ultracentrifugation.
This protocol is composed of established methods linked together for the quantification of small RNAs on highly pure HDL using high-throughput sequencing or real-time PCR. This video deals with the purification of the HDL.
This protocol is going to be demonstrated by my assistant, Evan Shidari.
To begin, add 90 microliters of 100X antioxidants to 9 milliliters of plasma. Then, adjust the plasma's density to 1.025 grams per milliliter by adding 0.251 grams of potassium bromide to every 9 milliliters of plasma. Rock the plasma at room temperature until all the salt has dissolved.